Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
In this video, we are going to discuss about extracellular matrix. So, extracellular matrix, as we discussed in the introduction and earlier classes, we know that for a particular under tissue triad also it will come as a biomaterial where we will be using we will be copying or mimicking the extracellular matrix. So extracellular matrix is usually it's a kind of interconnected network, so that will be present in every cells and every organ, so that which will act as a deciding factor for a particular structure, or even it gives the structural and mechanical support for the particular cells to grow on these particular matrix. So that they can adhere to the extracellular matrix, they can differentiate, and then they can and they can grow, and then finally it will be the tissue can be formed. So along with that, we are having biological active compounds or factors or growth factors that will be added so that the particular cells can grow. Okay, in this particular extracellular matrix. So we are having classifications also. It has got some components also, like insoluble ECM components and soluble components. All those things we will be discussing in this particular topic. So pay attention to this extracellular matrix, because this topic is actually repeated in many modules. I guess 2 to 3 modules this topic will be there, and even this module only for next 7th, 8th and 9th topic, even ECM can be written as one of the same answer can be written for the same for different topics. So tissue is made up of interconnected network of macromolecules known as ECM. So we know that what you mean by tissue, by the name itself, it is that it's like nothing but weaving. It's a French word which came from the weaving. Okay, the meaning is weaving. That means we are weaving the cells. That means like a kind of weaving the kind of network where the cells can attach to it, grow, and finally form a tissue. Understanding? So that itself we call it as a tissue. So now the tissue is made up of what? Interconnected network of macromolecules as extracellular matrix. So that's the definition. And ECM consists of mostly proteins and polysaccharides. So what does ECM consist, or is made up of? Proteins and polysaccharides. So ECM is synthesized, secreted, oriented, and as well as modified by the cellular components. Whatever the cellular components are there now, they will synthesize ECM. They can secrete the ECM, they can orient it and modify the ECM depending on what kind of organ we are talking about. Every organ inside our body has got extracellular matrix. Every tissue has got extracellular matrix. So the tissue, the extracellular matrix which is there for cartilage or bones, okay, is totally different for the soft tissues like ears and nose, or even the soft tissues. Okay, so there will be significant variations. So that's why this ECM it depends on what are the cellular components are there. Based on that, you will be having your ECM respectively. So in turn, ECM, that is extracellular matrix, controls the cell fate processes like adhesion, differentiation, migration, and apoptosis via integrins. So integrins, all of you know that cell surface receptors or a kind of proteins, right? So ECM can also control over few factors. For example, now cell you are going to see it in the ECM if you are in the tissue engineering. So the ECM, the cell has to come and attach to this particular ECM. So that is called as adhesions, right? The cell fates, in cell fate there are a lot of processes. That is adhesion, cell adhesion. Next, differentiation, that the cell undergoes differentiation to the next cycle, then followed by the different various cycles in the presence of growth factors that can be facilitated. And then the migration itself, how exactly the tissue is moving based on how exactly they are differentiated based on that. And then finally the apoptosis. You know what is apoptosis? It's a programmed cell death. So all these things comes under the cell fate. Okay, so the ECM can control the cell fate. Understanding? So ECM can control the cell fate processes like adhesion, differentiation, migration, apoptosis via integrals. So this ECM alone not, but through the integrins it will control. So the components of the ECM vary with the tissue type. So whatever the components are present in the extracellular matrix, that varies with the tissue type. That's what I told you. For cartilage, there will be a different ECM, where the composition will be totally different, and for soft tissue, like normal muscles and whatever the muscles we are having, like in biceps or in soft tissues, okay, there will be a difference, a significant difference in their properties or the components. So depending on the components, the ECM will vary to different tissue types. So for example, basal lamina that found at the base of the epithelial tissue, that consists of mainly collagen 4, laminin and heparin sulfates. These are the components of ECM, collagen 4, laminin and heparin sulfate, where when we are talking about the basal lamina, okay, which is found usually underneath or base of the epithelial tissue. So epithelial tissue always will have for a base, that is ECM, right? So this, in this particular tissue type, that is basal lamina, so the ECM components are different, like collagen 4, laminin and heparin sulfate. So on other hand, if you are comparing with the connective tissue, then the ECM consists of collagen 1. See, the difference is there it was collagen 4, here it will be collagen 1, and there it was laminin, here it will be fibronectin and elastin, and as well as glycosaminoglycans. So these are the different things. So here basically, here in this example, we are making out that the particular ECM for a tissue type, it will have different components. For basal lamina, it has got the components of ECM is collagen 4, laminin and heparin sulfate, whereas for connective tissues, the ECM's components will change, that is collagen 1, fibronectin, elastin and glycosaminoglycans. Next, the key components and the functions of the ECM. So ECM is nothing but it's a network of proteins and carbohydrates, or you can say the polymers that binds to cells together. So here the network will be there, like a mesh kind of thing you can imagine. The cells can be seated there. So here this network is made up of proteins and carbohydrates that will bind to the cells. That means cells will come and bind to this particular network, okay, or ECM. So that this ECM supports and surrounds the cells, like it will give the mechanical support. For example, if our ear is in this particular shape, the major contribution for this particular shape or morphological morphogenesis is ECM, that is extracellular matrix. So it regulates the cell activity. That means, as we just saw in the previous slide, that it controls over the cell fate processes like addition, migration, differentiation and even cell death, that is apoptosis. So it can regulate, in that way it can regulate the cell activity. Next, the pathways for cellular migration. It also controls, involves in the pathways of cellular migration, embryonic development, wound healing and management of this growth factors. So wound healing, all of you know, when there is a wound is formed, what happens when there is a wound, what happens after few days, we will get back those muscles and cells back, right? Only the scar formation will be there, but you will get the cells back. So that in there also the ECM plays a very important role, that will give the structural support there and as well as mechanical support. So next, management of the growth factors. That means how much growth factors are required for a cells to in order to differentiate in the particular ECM. So that also can be monitored by this particular ECM. That also depends on the ECM. So these are some of the key components and the functions of the ECM that you can remember. So the key components and functions here, one of the example I've given, since we discussed about two things, epithelial cells and connective tissue. So this is how the epithelial cells will be there. The basal lamina, this is where we can find the ECM, and then collagen. For connective tissue, we can find the different components like collagen fibers, fibronectin, okay, all these things, proteoglycans or glycosaminoglycans, all those things can be seen. Okay, this is for this diagram is just for your understanding purpose, how exactly the epithelial cells, here we discussed now the basal lamina and connective tissue. So basal lamina and connective tissue, how exactly they look like. Okay, in terms of ECM, the class of macromolecules that forms ECM. So we are having the components of the ECM. So those are called as macromolecules. So it has got two different distinct classification, insoluble and soluble components. So insoluble components, we are having kind of fibrous, collagen, elastic fibers, fibronectin, laminin, all these things comes under what you say, insoluble components. Here you can see all them, fibroblast is there, elastic fiber is there, okay, all these things are there, okay. And then for soluble, you have polysaccharides, polysaccharide chains, usually the proteins, and as well as combined with the polysaccharides or polymers, proteoglycans, next hyaluronic adhesive glycoproteins. Here you can see hyaluronic proteoglycans and glycoproteins. All these things can be the example for the insoluble components. So we have got both soluble and insoluble components. So soluble components, we will see that's a collagen is one of the example. That's a collagen is a strong resisting tensile forces and in abundance in Achilles heel. That is nothing but it will act as a natural polymer in short. So major insoluble fibrous protein of the extracellular matrix and the connective tissue is collagen, right? So most abundant protein in animals is collagen, and collagen made by the fibroblasts and some epithelial cells. So collagen structure, this is image is taken by the reference, okay. Here I have given the reference which was published in Protein Science Journal in 2002. So here this is the crystal structure of the collagen, triple helix model, gly, that is for glycon, and 10304. So that was the specific name is given to this particular structure, which was published in 2002. So types of the collagen, we are having different various types in the collagen. We are having fibrillars, sheet forming, connecting, and as well as transmembrane. So fibrillars forms structures such as a tendon or a cartilage, whereas the next sheet forming usually we require for many cells which are sensitive for that sheet forming is there. Collagen also provides sheet forming. And then connecting, it supports and organizes fibrous collagen, and then transmembrane also collagen can be formed. So collagen is a key component of wound healing. Obviously, whenever the wound healing is happening, the collagen plays a very important role. So collagen is the chief constituent of the ECM. That means out of all the insoluble and soluble components, collagen is a major collagen. You can say that collagen is a major part of the ECM or major component in the ECM. Okay, so chief constituent of the extracellular matrix protein in human body, which provides mechanical strength for various tissues and organs. So simply ECM functions, whatever we have seen, the same functions will be assigned to the collagen, because most of the ECM will be consisted by the collagen. Okay, so presently 27 types of collagen types are there, with at least 42 distinct polypeptide chains that have been identified so far, and still research and development is going on, and still we can get the more number of collagen types and distinct polypeptide chains. So next, moving on, so recent evidence indicates that the collagen and its derivative fragments control any cellular functions. So recently there is a study which says that the collagen and its derivatives, they can have various control over various functions, like cell shape, how the cell has to be shaped, and then differentiation also, it affects differentiation also, cell differentiation also, and migration, and as well as synthesis of proteins. So that was about collagen. Then speaking about elastin. So elastin chains are cross-linked together to form elastic rubber-like fibers, offers elasticity of the tissues. So that means elastin chains are cross-linked together. Okay, and we know that elastin is nothing but something which should have elasticity, right? In the name itself it is there. Elastic rubber-like fibers will be there, they offer elasticity of the tissues. Each molecule of elastin uncoils into more extended formation or conformations when the fiber is subject to stress. So whenever there is the subject or the particular molecule is subjected to stress, the molecules or the collagen present in those molecules, they will extend, they will uncoil into many more, the extended conformations. And when the when this particular force is removed, then it recoils spontaneously while stretching force is relaxed. So elastin is rich in hydrophobic, you can say that hydrophobic amino acids. It's synthesized as soluble tropoelastin, and it is a cross-linked by lysyl oxidase enzyme. So next we are having fibronectin. So it's a dimer that is composed of two very large subunits joined by the disulfide bonds at one end, and each subunit is folded into a series of functionally distinct domains. So all form of the fibronectin are encoded. Fibronectin binds integrins. Those are nothing but what? Surface receptors. So fibronectin secreted primarily by the fibroblasts. So cell division, cell adhesion, cell growth, migration and differentiation. It helps in all these things, and and fibronectin is very important for wound healing, and also fibronectin is used for cancer and as well as fibrosis treatments. So that was about insoluble components. I guess there was a confusion in the heading. Yes, here it should be insoluble, right? See, we discussed collagen, elastin, fibronectin, right? Those are nothing but insoluble. Apologies for that. Maybe typo. So that was about insoluble components of these things. So we discussed about collagen, we discussed about fibronectin and also elastin. Okay, the three components we discussed. So next, moving on to the soluble components of ECM. So we are having usually the proteins and polysaccharides. They comes under the soluble components of the ECM. So first one is we are having GAG. So that is nothing but glycose aminoglycans. So these are usually we can also call them as a polysaccharides. They are a form of the type of polymer. So they belong to polysaccharides. Okay, it is the most abundant. This GAG is the most abundant heteropolysaccharide. You should remember, when you are talking about the most abundant polymer, then it's collagen. When you are talking about most abundant heteropolysaccharide, then it is GAG in the body. So it is the long unbranched polysaccharides comprising of repeating units of the disaccharides, either this GalNAc, that is nothing but N-acetyl galactosamine, or N-acetyl glucosamine, galactosamine or glucosamine. So with uronic acid, that is very important. You should mention with uronic acid, that is nothing but glucuronate or iduronate. Okay. So the high negative charge molecules provides the extended conformations which imparts the viscosity of the solution. So whenever there is a high negative charge of this particular molecule is there, so that means that will give you the extended conformation which imparts the viscosity of the solution. That means whenever the solutions are there inside our body, if viscous, it has to be viscous, then it will incorporate that viscosity. Now the higher the viscosity and lower compressibility, that provides the lubrication at the joints. Usually we experience in the all of our joints, we need more viscosity. That means more viscous in the sense more cushioning effect, less viscous in the sense less cushioning effect or less lubrication effect. So examples includes hyaluronic acid, then dermatan sulfate, heparin sulfate, keratin sulfate. All these things will come as an example for this particular GAG. So proteoglycans. Proteoglycans are proteins that are heavily glycosylated. That's why they are called as proteoglycans, because they are glycosylated heavily. Now the basic proteoglycans unit consists of a core protein. Definitely the protein will be the major part, and then other part will be the glycans. So with one or more covalently attached glycosaminoglycans, that is nothing but GAG. So we are having the GAG also in proteoglycans, right? The protein core of the proteoglycans allows the multiple GAG attachments. So a number of GAG can come and attach to the proteoglycans. So that was about the soluble and insoluble components of ECM and the introduction to ECM. Okay, I hope you understood. Okay, next topic we are having cellular components that we will discuss in the next class.